All right, great. So what I'd like to tell you about today is our work on translating genomics into therapeutic insights. And specifically, how do we dissect the circuitry of human disease in order to manipulate it ultimately at single cell resolution. So I'm a professor of computer science at MIT. I'm a member of the Computer Science and AI Lab and also a member of the Broad Institute. You can find out a lot more about our work on this website and contact me through email or Twitter or any other way. So why are we here? We're here because genomic association and genetic association studies have truly revolutionized the way that we understand human disease. We are able now to, in a completely unbiased way, discover all of the genetic loci that are predisposing us to different diseases by carrying out large-scale genome-wide association studies. What these studies reveal are the genetic associations, in this particular case with body mass index, a measure of obesity, across the entire genome, where every SNP is a dot, every single nucleotide polymorphism is a dot, and the height of that dot is the probability or the negative log 10 probability that this association is fortuitous. In other words, the higher the dot, the more unlikely, the more non-random, the more significant the association is. And in this particular case, this genetic association study back in 2007 revealed that FTO, this genetic locus here on chromosome 16, is the strongest genetic association with obesity. What does that mean? That means that we now have the ability to understand the mechanism underlying this region and ultimately reveal new target genes, new therapeutics, and enable precision medicine and one day personalized medicine. The challenge, of course, is that understanding the mechanism is not simple. Genome-wide association studies have the advantage that they will reveal an association no matter what the mechanism is. But the disadvantage is that when you look under the hood, you actually have no idea what the mechanism is. For 93% of cases, these disease hits are non-coding. They lie somewhere in the non-coding genome. And here you would say, well, it's right in the middle of the FTO gene, but it's in the middle of the first intron of the FTO gene. The second exon is there, the third exon is there, the first exon is, far, far away. So in this very long first intron, there are 89 common variants, none of which is actually disrupting the protein coding region for FTO. So that means that we actually don't know the target gene. We don't know that it's FTO. In fact, what we showed in our own work, published just a few years ago in the New England Journal of Medicine, is that the true targets of this genetic association are in fact sitting 1.2 million nucleotides away and 500,000 nucleotides away. So that basically means that we really have no clue whether this is in fact the true target gene when we see genetic associations that fall even within a single gene. And of course, when you have 5 or 6 genes within the genetic locus, you really have no idea. And it could be any of these 5 or something far, far away. And that's just the beginning of the adventures, finding the target genes. You also don't know the causal variant, which is very important for establishing causality. In this particular case, we showed that it was RS1421085 sitting right there. And the reason is that we found that it overlaps a massive super enhancer that is disrupted by that genetic variant. We also don't know the cell type of action for these regions. It could be acting in the brain, it could be acting in the lung, in the liver, anywhere. And what we showed in this particular case is that it was acting specifically in the first 3 days of differentiation of adipocytes when they're making the decision to go between white fat and brown fat. And I'll come back to that later. We also don't know the relevant pathways. We don't know the mechanisms and so on and so forth. So what I'm going to tell you today is how do we translate the promise of genetics into a delivery of mechanism rather than just association. So the problem with giving a talk over Zoom is that I can't say, hey, who's with me? So what I'm going to do is ask people to use the little reaction button out there and then give me a thumbs up or, this is awesome. This is great. I see some thumbs up. That's great. So if you guys don't mind, you can also turn on your videos and give me like visual feedback as to or something like that. Because, we're all human and seeing other humans is kind of nice. So thank you, Will. Thanks, Jonathan, for turning on your videos. Thanks, Aleja. This is awesome. All right, cool. So again, I'm going to be introducing a lot of new material. So the more people I can actually see the faces of, the better. Let me hide this again. Awesome. So the goal is understand genetics. The challenge is that the vast majority of it is non-coding, and the answer is figure out the circuitry. So how do we do that? We basically integrate both common and rare variants across many genetic associations. Here I'm showing you Alzheimer's instead, where the ApoE locus reigns supreme, and there are dozens of other associations. We basically start with genetics, and then we profile RNA and the epigenome across both healthy and disease samples. Why do we do that? To understand how genetic variants are influencing these intermediate molecular phenotypes. To also understand how they're influencing expression and what are the target genes through which they're acting. To also understand the cell type where they act. So we do this across healthy individuals to understand the impact of genetic variation, but also in disease individuals to understand how are non-genetic factors, including environmental factors, influencing ultimately the impact on disease. And why are we looking at the epigenome? Because the epigenome gives us a glance, a lens to look into the circuitry of disease, to basically understand what are the control regions that govern the expression of genes. And these control regions are basically marked by, number one, DNA accessibility. Number two, they're marked by specific histone modifications that are associated with enhancers, promoters, transcribed regions, the basic building blocks of gene regulation, because they remember the memory of activity of different regions. And therefore, to remember the identity of the cell types, you need to mark the regulatory regions in particular ways. And by looking at these marks, we're basically looking at the notes that the genome is taking to remember where are the control regions. And we're also looking at DNA methylation, a mark of repression in regulatory regions, but also mark of activation in transcribed regions. So we're profiling RNA and epigenome systematically across thousands of samples, hundreds of tissues, dozens of individuals per condition, and both healthy and disease samples. We then integrate the data to predict driver genes, regions, and cell types. And then we validate our predictions using human cell cultures and mouse models and any other sort of relevant systems. And of course, disseminate the results and start all over again. So what does that give us? That gives us circuits instead of regions. What genome-wide association studies give you is a region. Something is going on here. What we can understand now is circuits, namely, what are the specific genetic variants associated with this region? What are the control regions where these variants are occurring? What cell types are these control regions active in? What are the upstream regulators that control these regions and whose motifs are sometimes disrupted by these nucleotide variants? And what are the target genes and ultimately the function of these regions? Who's with me so far? Yes, awesome, good. So looking back now at the previous slide of this FTO locus, what we basically showed is that the FTO gene itself has nothing to do with the association. Instead, we can narrow down the association to a single nucleotide alteration that changes this AT-rich motif by changing the T into a C, disrupting the binding of ARID5B, which can no longer repress this massive super enhancer, which then leads to overactivation, a gain of function of these two downstream target genes, IRX3 and IRX5. IRX5 is 1.2 million nucleotides away. IRX3 is 500,000 nucleotides away. We then figured out what these genes do. And what we found is that they are master regulators of a process known as thermogenesis, or heat generation, within our fat cells by depolarization of the mitochondrial membrane, causing a loss of the proton gradient and loss of energy as heat, rather than storage of excess energy in white adipocytes to be used later. So that basically leads to risk individuals, including myself. So I'm homozygous risk for the obesity allele. So it causes us risk individuals to basically be unable to burn excess calories. And instead we end up storing them all. And it takes us from a predisposition to be lean to a predisposition to be obese. Great. So why do we do all this? Just to tell people bad news? Of course not. In order to understand the circuitry, we can then manipulate the circuitry. And knowing these additional knobs, knowing the upstream regulator, knowing the cell type of action, knowing the downstream target genes, and knowing the causal variant allows us to now start intervening. So we can develop new therapeutics for manipulating the upstream regulator, the regulatory region, the downstream target genes, or any of these processes that we are finding at the cellular level. And to demonstrate causality of this locus, we basically went through the steps of perturbing each of these knobs and finding that, in fact, each of them switches like a switch between lean and obese phenotypes. So disrupting a single nucleotide variant out of 3.2 billion nucleotide variants in the human genome basically leads to restoration of the cellular phenotype of thermogenesis in primary adipocytes from risk individuals. So we took primary adipocytes from homozygous risk individuals, and then we found that indeed they're unable to thermogenize, measured as relative oxygen consumption rate. We then stimulated them, and again, nothing happens in the risk individuals. But if you use CRISPR-Cas9 genome editing to alter a single nucleotide and restore this AT-rich motif so that the ARID5B repressor can bind that super enhancer, you basically see restoration of thermogenesis, both in basal as well as in asparterinol. We have a 7-fold increase in thermogenesis here using a single nucleotide alteration. I mean, that's precision medicine right there. Of course, therapeutically, you wouldn't edit the genome of individuals, you would manipulate that circuitry. But for causality demonstration, we need to show that the SNP is in fact responsible for that association. We then basically said, okay, what about manipulating the downstream target genes? Can we have an organismal effect, not just a cellular phenotype effect? And indeed, when we overexpress a dominant negative form of IRX3, thereby repressing these overactivated genes that lead to obesity, by overexpressing them, they would basically be leading towards the lean condition. So basically, by repressing IRX3 using this overexpressed dominant negative form, by repressing it, you would basically shut off this repression and in turn turn on thermogenesis. And this is exactly what we find. So basically, we find that mice basically burn all of their fat stores. If you look at their fat stores, they're completely depleted. They are simply burning all their calories when they're awake, when they're asleep. And if you put them on a high-fat diet, normal mice gain weight. These mice are just simply unable to gain weight. They're simply burning all these calories. They don't change their exercise. They don't change their appetite. They simply burn all these calories. So how do we do this systematically? What are the tools that we're using to uncover these circuits and to manipulate the circuits? So first, we're building reference epigenomes. Then we're looking at how these epigenomes change from individual to individual. Then we're looking at this at single cell resolution and understanding the cell type specific genetic effects, then going across multiple phenotypes, multiple tissues, and lastly, developing methods for high throughput dissection. So diving into the reference epigenome building, we basically have been part of several large-scale projects, including the ENCODE project, the Roadmap Epigenomics project, EpiMap, published more recently, and this EpiAtlas that we're now involved in as part of the International Human Epigenome Consortium, to basically profile adult, embryonic, and induced pluripotent stem cells, embryonic stem cells, in vitro differentiated cells, and primary cells from a diversity of human tissues to characterize systematically the epigenomic landscape by measuring promoter-associated marks, enhancer-associated marks, transcription, polycomb repression, heterochromatin, activation of enhancers and promoters, DNA accessibility, DNA methylation, and what I like to call as the output of RNA expression. So we basically systematically characterize all these different facets of the human epigenome, and then we're integrating all these combinations to define proximal regions known as promoters that are marked by K4M3, distal regulatory regions that are extremely cell type specific that we call enhancers, transcribed, repressed, and other regions using specific combinations of these modification marks. What this gives us is a picture of the human epigenome across all 3 billion nucleotides and across 127 different epigenomes here. That allows us for the first time to characterize systematically the non-coding landscape, the circuitry of these cells. And in particular, we can look at how promoters are very stable, how enhancers in orange are very dynamic, how transcribed regions turn on and off, how some genes are extremely repressed across all of the cell types, except for a handful where they appear to be de-repressed, and how non-coding regions are acting. So for example, if there's a genetic association with disease here, I can basically say, well, what are the regions where these non-coding region might be active without having to look at the genes nearby? And once I have these, I can also start asking what could be target genes. And instead of only looking at the genes nearby, I can say, well, hey, this PAX5 gene here is in fact co-expressed with whenever this regulatory region turns on. So maybe this is the true target of that region, even though it's several genes over in this annotation. So we now have a handle to the circuitry of disease. And we can basically start asking, where are the genetic variants associated with different traits located? So we can find the genetic variants associated with height, paint some subset of the human genome. Those associated with type 1 diabetes, a different subset. Blood pressure, a different subset. So for all of those, we can find the region of association, shown here as a bar, and then these tall bars showing the individual nucleotides within them. So we can now start overlaying these regions with annotations of active regulatory elements, active enhancers in different cell types, in stem cells, in immune cells, in heart cells, in liver cells, and so on and so forth. What that allows us to do is now start asking, is there an enrichment? Are genetic variants associated with height preferentially overlapping enhancers active in different cell types? And this is indeed what we find. If you look at genetic variants associated with height, they're only enriched in enhancers that are active in embryonic stem cells. Immune-associated traits are, in fact, overlapping enhancers active in T cells and B cells. If you look at blood pressure-associated variants, they're overlapping specifically enhancers of the left ventricle. This indicates that genetic variants associated with blood pressure affect not the genes, but the control regions of the left ventricle, because this is looking specifically at enhancers, at non-coding regions. If you look at genetic variants associated with fasting glucose-related traits like type 2 diabetes, you again find a very specific association with enhancers of pancreatic islets. Genetic variants associated with cholesterol, HDL, LDL, and so on and so forth, specifically associated with liver enhancers. If you look at inflammatory bowel disease, you basically can expect to see association with immune cells, given the inflammatory component, and of course, digestive tissues, given the bowel disease component. And indeed, this is what we find. If you start now making guesses for Alzheimer's disease, you would have expected to see the brain. And when we look, we find no association, no enrichment in the brain. What does that mean? Well, it's a little strange, something could be wrong, but in fact, we find an enrichment specifically in CD14 plus cells, a mark of monocytes, macrophages, and the immune cells of the brain, known as microglia. But microglia compose a very small fraction of the brain cells. The vast majority of brain tissue is excitatory neurons, inhibitory neurons, oligodendrocytes, astrocytes, and only a tiny minority is in fact microglia. So this suggests that perhaps genetic variants associated with Alzheimer's act first in the microglia and only later have impact in the rest of the tissues. So to test that hypothesis, we basically sorted neurons, oligodendrocytes, and microglia from brain, and we profiled H3K27 acetylation using chromatin immunoprecipitation. And indeed, we found that the genetic variants associated with Alzheimer's using LD score regression localized specifically within the microglial enhancers. And indeed, if you look at the progression of Alzheimer's disease in a mouse model developed by Li Hui Tsai at the Picker Institute, you see that early in Alzheimer's disease, you see changes that implicate immune processes, but not neuronal processes. You only see these changes happening later during the progression of Alzheimer's disease. So that suggests that, in fact, there's an immune basis of Alzheimer's, both in mice and in humans. We have since expanded from 127 epigenomes to 834 epigenomes in a paper that we just published a few months ago. And this took us from 54 enriched GWAS traits to 534 enriched GWAS traits, looking here at this incredible diversity and painting them together in a network that basically tells us that, for example, schizophrenia, depression, worry, neuroticism, etc., localized in brain, where again, they likely affect primarily the neurons, similar to educational attainment, intelligence, cognitive performance, and so on and so forth. By contrast, Alzheimer's, as I mentioned earlier, is localizing instead with immune cells. Kidney, for example, shows associations with filtration traits, liver with cholesterol, heart with a lot of heart repolarization, blood pressure, etc., as we saw earlier. But now you can also see that every one of these is a pie chart that is basically telling us what is the association of each trait with a diversity of tissues. For example, coronary artery disease is one of these most pleiotropic traits that appears to be acting in enhancers from many different types of tissues. The top associations are with liver, coronary artery, thyroid gland, adipose, and so on and so forth. What we can now do is start partitioning that complexity, start asking, if I take the genetic associations of coronary artery disease, and I ask what subset overlaps liver enhancers, what we see is that the biological processes associated with those are dramatically different from those that overlap instead coronary artery, and so on and so forth, enabling us to start partitioning the diversity of traits, the diversity of individual molecular phenotypes that coronary artery disease is based on through the different tissues where these genetic variants are acting. And we can do this also at the level of comorbidities and co-associations with other traits. So those genetic variants show very distinct co-associations depending on the tissue that they overlap. And we can do that at the level of individual loci by going through all of the genetic loci associated with coronary artery disease, and then asking, do they overlap coronary artery enhancers or liver enhancers, and so on and so forth. So here's a handful of examples where PCSK9, one of the strongest genetic associations, one of the strongest surprises and successes of genetic studies, is in fact localized very specifically in liver enhancers and is linked to the PCSK9 gene. So you can see here the genetic variants in a GWAS zoomed in plot here are in fact linked to the promoter of the PCSK9 gene. Similarly, for eDNRA, this locus here, you see all of the genetic variants are in fact forming two distinct associations, both of which appear to be linked to the promoter of eDNRA, which is acting in heart. By contrast, if we see this PLPP3 locus, which is slightly higher ranked, we basically see that there are multiple genetic variants overlapping multiple enhancers, and these are linked both to liver and to heart, two different genes that are sharing this regulatory control region here that are divergently transcribed and that are acting in heart and in liver, suggesting perhaps that there's multi-gene and multi-tissue pleiotropy in this locus, indicating that perhaps it's acting through multiple mechanisms. So we basically built a resource for doing this systematically at compile.mit.edu/epimap that allows you to look at the linking of enhancers to target genes, to visualize the SNPs in 30,000 loci, along with the top most enriched tissues and their likely target genes, and then understand the gene regulatory circuitry between transcription factors, enhancers, motifs, and ultimately target genes. But the epigenome is only one way to annotate the genome systematically. We can also use comparative genomics to do this annotation. So we have previously developed in a series of papers in Nature and other places, looking at evolutionary signatures for systematically annotating protein coding genes based on their distinct patterns of evolution, RNA structures, microRNAs, regulatory motifs, and so on and so forth. This allows us to now start revisiting the genome. For example, we found hundreds of new protein coding genes and new protein coding exons, and 118 of those actually overlap genome-wide association hits that were previously thought to be non-coding, but turns out to be protein coding after all. Here are some examples associated with all kinds of biological processes, like trafficking or leukemia or cerebrum and so on and so forth. And these include, for example, some quite striking examples. This genetic association with myopia was previously lying in a non-coding region, well upstream of this TJP2 gene. But what we found is a new protein coding exon, which we now realize is actually expressed very specifically in the retina. And this SNP is in fact perturbing that protein coding exon directly. So that basically suggests that maybe we were being a little myopic about this myopia association. And instead of having to look for non-coding regions, we can in fact now use these new protein coding annotations, which provides a new mechanistic insight about the target gene and the likely tissue of action and the function of this region. So there are many additional such examples of being able to interpret new genetic variants that were previously unaccounted for using protein coding associations. But we also can use comparative genomics to pinpoint individual regulatory motifs and start looking for genetic associations within these regions. So one example is this genetic association here, which in fact disrupts this genetic variant, disrupts this motif for this FOXO regulator, and it is acting specifically within this immune enhancer, suggesting that we may have, again, candidate functions for these SNPs. We can also use comparative genomics in the current pandemic to start understanding the genome, not just of human, but also of the SARS-CoV-2 virus responsible for the COVID-19 pandemic that has us all here, sitting here over Zoom instead. And what we can do is start asking, can we use these evolutionary signatures to annotate these 29,000 nucleotides? And what we find is that, for example, ORF10, which was previously annotated as protein coding, is in fact not showing signatures of protein coding constraint at all. By contrast, we can discover a new protein coding gene, ORF3C, within ORF3A. So even within a genome as small as SARS-CoV-2, we can still discover new protein coding genes. We can also use comparative genomics to study the evolutionary rate of these genomes across multiple distantly related strains, and also within the current pandemic, to basically show that, for example, the spike protein S1 is evolving slower than what we would have expected, but the nucleocapsid protein is evolving much faster than what we would have expected. And in fact, we find that the fastest evolving region of the genome of SARS-CoV-2 is in fact within antigen region where an epitope is disrupted, likely to evade the human immune system. Lastly, we can start interpreting these mutations that are now rising to high frequency in many different cities. Here's an example of D614 gene, one of the early D614G, one of the early mutations that rose to high frequency independently in many different cities. And we can now see that this is disrupting an 11 amino acid region that had never been changed in any amino acid whatsoever, and disrupting a nucleotide that itself had never been changed across all of these related genomes. So you can use now this reference map of the human genome and the human epigenome to start asking, how are these regions changing in the context of disease? So we're now going to be profiling not just a reference epigenome, but many variants of the epigenome across different individuals, relating that with genetic variation and relating that with expression variation in the context of disease. The goal is to bridge the gap between genetic variants and disease. This is a very large path, and each of these variants contribute an infinitesimally small amount to disease. So we have millions of these genetic variants, any one of which is having a tiny little effect on disease. But by profiling specific tissues, specific regulatory regions, specific genes, and specific intermediate phenotypes, we can actually start mapping the correspondence between the two and start painting a path of causality between a variant, the tissue where it acts, the regulatory region, the gene, the intermediate phenotype, and ultimately disease. The challenge, of course, is that the environment might also be contributing both to disease and to these intermediate variables. And also some of these genes might be changing in response to disease or even to combat the disease phenotypes. So what we're doing here is combining genetic variation, epigenomic variation, and phenotypic variation together to first understand how we can predict the epigenome from the genome using methylation quantitative trait loci, or QTLs, then combining the two to start evaluating the causality of genetic variants that are mediated through the epigenome or the transcriptome. And we're doing this in the ROS-MAP cohort, the Religious Order Study in Memory and Aging project that David Bennett put together at Rush University many, many years ago, and in collaboration with Phil DeJager, previously at HMS, now in Columbia. We basically discover 50,000 genetic loci where, if I know your genetics, I can predict your epigenome in your brain in an inaccessible tissue at 93 years of age. This is quite remarkable. It basically means that for many of these loci, I can actually start imputing your methylation profile from your genetics. That allows me to now not just carry out a genome-wide association study that basically has this very long arrow, but also a methylome-wide association study that's looking at the methylation changes and their correlation with disease. But that is a bidirectional arrow because it's not coming from genetics directly. Instead of doing a methylome-wide association study, I can use the methyl QTLs to predict the methylome. So get an imputed methylome, which is genetically driven. And now, not just look at methylation, but look at the genetic component of methylation and how it correlates with disease. That allows us to now start predicting a path of causality once more, because it's the genetic component that I can predict at birth. And that causality allows us to now apply these associations to many, many more individuals, because we only need genetics from these individuals. We don't need to profile their brain directly, and therefore we can expand to many more individuals. Using this approach, we can now find that in chromosome 6, for example, that has no genome-wide significant hits, we can now find several genome-wide significant hits because we now are testing fewer hypotheses. So we're finding many additional loci that are now robustly and significantly associated with Alzheimer's disease. And we can do this not just with methylation, but also with transcription, with disease, to identify additional loci across multiple layers of regulation. In particular, we can go to regions of genome-wide significance, shown here in purple, as well as regions that are not previously genome-wide significant, shown here in gray, and find the directionality of effect, the target genes, and the likely mediating tissue of these associations, discovering many new targets and many new loci associated with disease significantly. And you can see in here the size of the circle represents the fraction of heritability captured in each of these loci. So this is basically giving us a path of causality by combining genetics, epigenomics, and transcriptional variation in the context of disease. But it is working at the bulk level. And we saw earlier how in brain, there's very specific subtypes of cells that are associated with disease. So we basically turn to single cell dissection of these epigenomic and transcriptional changes. We have now profiled more than 1,500 human postmortem brain samples across 20 million plus cells in Alzheimer's disease, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar disorder, Down syndrome, and several other psychiatric and neurodegenerative disorders. In each of these traits, we're basically looking at about 48 individuals across many different individuals, many cell types, many cells, multiple brain regions, and looking at both RNA and DNA accessibility at single cell resolution. In the first paper that we published about 2 years ago in collaboration with Li Wei at the Picard Institute, we basically looked at 48 individuals, including 24 non-AD individuals, some pre/mild AD, and some severe Alzheimer's disease. And you can see here the phenotypic variables changing from high cognition to cognitive loss, and from low pathology to high pathology, including plaques, neurofibrillary tangles, and so on and so forth. The cells from these individuals cluster by cell type, and we are able to now start asking within each of these subtypes of cells, what are the phenotypic enrichments when we start subdividing excitatory neurons, inhibitory neurons, astrocytes, oligodendrocytes, OPCs, and microglia. And what we're finding is that subsets of excitatory neurons are associated with high or low amyloid pathology, with advanced or early BRAC stages of disease progression, with advanced or low cognitive decline, and with AD or non-AD. Moreover, the subsets of cells that are associated with this more severe disease are in fact more frequently associated with female individuals as well, suggesting that at the transcriptional level, female individuals show stronger transcriptional pathology. We're also able to distinguish early changes versus late changes, and what we're finding is a lot of cell type specific processes changing in Alzheimer's. But in late disease progression, we're finding a lot of common changes, many of which are in fact associated with damage response, including protein folding, DNA damage, and so on and so forth. We're finding that the number of sex-specific expression genes are in fact greatly increasing with Alzheimer's. We have 3,000 genes differentially expressed between men and women before AD, but 6,000 after AD. And this is true for many of the cell types. And in particular, we found that these genes that are differentially active in Alzheimer's show sometimes sex-specific biological enrichments. For example, we found myelin-associated genes in oligodendrocytes are upregulated in men, but not in women. And indeed, we found that there's increased white matter loss in female individuals, suggesting that perhaps we should be thinking about sex-specific treatments in addition to stage-specific interventions. We have since expanded to many more brain regions, looking at hippocampus, thalamus, mammillary body, entorhinal cortex, angular gyrus, midtemporal cortex, and prefrontal cortex that I showed you earlier, and looking at 48 individuals for each of those. This gives us a very large number of cells that we can now start subdividing into many different subclusters of excitatory, inhibitory, astrocytes, and so on and so forth. We find 30 different subtypes of excitatory neurons, and here's the corresponding marker genes, and 23 subtypes of inhibitory neurons. We're finding that these are changing dramatically from region to region. You can see here in the anterior thalamus, very specific subtypes of inhibitory neurons, changes in the hippocampus versus the angular gyrus. Similarly, astrocytes change them dramatically between different brain regions. You can see here the thalamus has a very distinct set of subpopulations from mammillary body, from these cortical and subcortical regions. We also are able to take these 1.6 million cells across these many individuals and start predicting their association with Alzheimer's disease by building a machine learning model that allows us for every cell to predict the transcriptome-based signatures of Alzheimer's at the cell level. That allows us to now start ordering the cells based on whether they show early signatures or late signatures of healthy versus Alzheimer's disease. And we can see where the expression of each gene is changing relative to this time course of Alzheimer's progression. So this allows us to distinguish early acting versus late acting genes and the cell types and tissues where they appear to be acting. We also are able to use these data to start understanding the sub-regions, the anatomical subsets of the hippocampal formation in this example, looking at dentate gyrus versus CA1, 2, 3, subiculum, and the entorhinal cortex layers in this particular case, and how the single cell expression patterns allow us to actually subdivide these neuronal cells into the specific subregion, an anatomical subregion where they're coming from. And in particular, we're finding that genes associated with CA1 expression, CA2 expression are much more predictive of AD than genes expressed in the dentate gyrus, suggesting that these regions are perhaps more severely affected. We're also using these single cell profiles to start predicting the three-dimensional localization of each cell based on its expression profiles. Na and Juna in our group have basically co-developed this deep learning model that allows us to go from coupled, paired single cell expression and localization of these regions transcriptionally using data that Xiao Wang at the Broad Institute generated, and placing not just neuronal cells, but also glial cells into specific three-dimensional locations using their single cell profiles by training a model that allows us to infer their spatial coordinate localization. And what we're finding is that both oligodendrocytes and astrocytes are in fact showing expression patterns indicative of where they are placed in these layers, and now enabling us to start asking for correlations between the changes in expression patterns of neighboring cells in the three-dimensional localization, not just cells in some sort of t-SNE space. Kai Kang in our group also developed methods for deconvolving these mini bulk expression patterns from Visium and other spatial transcriptomics profiles using our single cell reference profiles, and then looking at how this in fact reveals that there are a depletion for Purkinje cells in the cerebellum associated with SCA1 ataxia, when in fact the marker gene expression is not changing significantly, indicating that deconvolution is quite important in distinguishing the cell state, not just the expression of a marker gene. Expanding beyond Alzheimer's disease, we've basically started looking at many other psychiatric and neurodegenerative disorders. So here's an example from schizophrenia, looking at all of the different subtypes of excitatory and inhibitory neurons, and being able to distinguish the genes that change in expression associated with specific genetic loci associated with schizophrenia, and the cell types and tissues where these appear to be acting. In this particular case, we're able to see increased expression and decreased expression genes in red and in blue, and the specific excitatory and inhibitory neurons, as well as, in some cases, the glial cells where these change in expression. And these are linked using high-C evidence of genome folding in each of these cases. We're also able to predict from the transcriptional signatures of individuals schizophrenia versus control expression patterns. And this allows us to now start classifying individuals as to how schizophrenia-like are their expression patterns at single cell resolution in each of these cell types. What we're finding is that indeed the classification works very well for many individuals, but there are some exceptions. For example, this control individual is showing transcriptional signatures that are very schizophrenia-like. And indeed, his son is in fact a schizophrenic patient. These other individuals here are showing schizophrenia signatures at the phenotypic level, but control signatures at the transcriptional level. And again, all of these individuals are in fact showing an enrichment of this SCTR cell state. We're also able to find upstream regulators that are controlling these differentially expressed genes and group them into modules. And some of these modules are in fact acting both early in development and late in the adult phase. And they're implicating neurogenesis as well as synaptic organization and synaptic signaling. For psychosis in AD, we're basically finding this association with repression of neuronal activity. We're finding thousands of genes that are repressed in the hippocampus and the prefrontal cortex of AD individuals that are showing psychosis compared to individuals that do not show psychosis. And you can see these dramatically different expression patterns from both the hippocampus and the prefrontal cortex. The genes that are increasing or that are decreasing in expression in this particular example are associated with ribosomal biogenesis, signaling, neuronal projection, autophagy, apoptosis, secretion, and so on and so forth. We're also finding a lot of changes in ALS and FTLD, specifically implicating these long-range projection motor neurons, these Beck cells. And we're also finding that in Huntington's disease, we're losing the identity between these direct pathway versus indirect pathway spiny projection neurons that are quite distinct before Huntington's, but are losing their identity after, indicating again their importance in the disease. We're able to subclassify in microglia that were previously hard to even detect at bulk resolution. We're now able to find pro-inflammatory versus synaptic versus many different subtypes of microglia. And we're finding that they are associated with very distinct biological processes and also with very distinct associations in schizophrenia versus Alzheimer's disease. We've also just posted the first map of the brain vasculature at single cell resolution in human, looking at this gradation between arterial, capillary, and venule. And these zonation differences are in fact implicating these gradients in endothelial cells, in smooth muscle cells, as well as in pericytes. We're finding a new subtype of fibroblast that is specifically to human, specifically found in human, but not in mouse. And we're also finding these vasculature coupled cells that are pulled down with the vasculature and that are showing signatures of both the original cells and these vasculature cells, but are very distinct and do not represent doublets. And these are a very small minority of the overall cells, but when we visualize them, we find that indeed they are vasculature coupled. We've also linked non-coding regions to their target genes using single-cell correlation between DNA accessibility and RNA expression. We're finding that, for example, increased accessibility is associated with increased expression for ApoE in microglia. Similarly, decreased accessibility associated with decreased expression for RLPB1 in astrocytes. And if you look across many different genes, you see that there's these global correlations, global agreement between DNA accessibility changes and RNA expression changes, but not always. And there are some interesting exceptions. We're using now our single cell attack profiles to basically fine map non-coding variants associated with psychiatric and neurodegenerative disorders into specific SNPs that are localizing in these cell type specific enhancers that are enriched in these different disorders. Once more, we confirm the association of Alzheimer's genetic variants with microglia rather than neurons, indicating again an immune basis. And we're also able to predict the regulators upstream of many of these changes, including SPI1, a master regulator of immune cells, also known as PU.1, which is found in many GWAS loci associated with Alzheimer's disease. We're also able to go down to single cell resolution for somatic mutation detection, looking at the mutations that are accumulated by individual genes in specific cells. And what we're finding is that these disease-associated neurons using SMART-seq whole transcriptome sequencing are in fact showing increased mutations compared to excitatory and inhibitory neurons, and that glial cells that are not post-mitotic are in fact showing again more mutations, as you would expect given more cell divisions. We're finding that these mutations are enriched in Alzheimer's individuals and in female individuals, and we're able to start pinpointing individual genes and pathways that are showing an accumulation of this somatic mutational burden. Combining single cell and bulk profiles, we can now go back to GWAS and QTLs and start asking about cell type specific genetic effects by looking at how these bulk samples are deconvolved. And we're able to use that to now start pinpointing genetic changes associated with Alzheimer's disease are once more affecting the expression of genes in microglia for about 50% of cases and only a minority in other tissues and cell types. Looking at the different subtypes of Alzheimer's, the different signatures of amyloid beta and neuritic plaques, tau and neurofibrillary tangles, neuroinflammation, we're able to now start partitioning the genome according to the regions whose methylation correlates with specific phenotype signatures of Alzheimer's. And these regions are in fact showing enrichment in specific tissues and cell types and biological pathways, suggesting that perhaps we can start partitioning the complexity of these complex traits, not just in terms of their epigenomic enrichment, but also in terms of the specific cell types where they might be acting. Expanding beyond just Alzheimer's, we're basically looking at electronic health records across a diversity of disorders, looking at hierarchical models that are able to capture multimodal information from patient records, including prescription, doctor notes, ICD-9 codes, lab tests, and so on and so forth, and start classifying patients into subtypes that are including multiple of these different data modalities. Using this information, we're able to now classify patients of post-traumatic stress disorder, which has a strong environmental component, to start asking for subsets of these patients that we can then associate with genetic variants that indicate expression changes in particular tissues and cell types. Beyond individual tissues, we're able to look at multiple tissue changes that are coordinated in the context of exercise and obesity, both exercise versus sedentary lifestyle, and also high-fat diet versus normal diet. And looking at the changes that are happening in these thermogenic tissues of muscle and fat, as well as small intestine, cortex, liver, heart, and blood, in a large collaboration with Novo and with Harvard Medical School. We are able to annotate many of the cell types and subtypes, including a new subtype of these muscle progenitor cells, multiple subtypes of adipose stem cells, many subtypes of immune cells, and see how these are changing in the context of obesity and diet interventions. And what we're finding is that the two primary cell types that are changing are these adipocyte stem cells and these regulatory cells, T cells, that are in fact showing the strongest changes with obesity, that are basically showing this dramatic increase in expression and in cell-cell communication. And we're systematically finding that exercise is in fact reversing the effects, the detrimental effects of obesity across all of these different cell types. And we're also, again, once more pointing to adipose stem cells, these early stages of adipocyte differentiation, similar to what we saw with FTO, are in fact the most changing and likely to be extremely important in both within tissue signaling and cross-tissue signaling, along with these T cells. In the context of cancer, we're finding that there's a lot, we're finding biomarkers in cancer, in aging, in Alzheimer's. In the case of Alzheimer's, we're basically finding blood biomarkers using monocyte H3K27 acetylation, and are able to predict Alzheimer's disease using this signature, including for genes that are previously implicated with Alzheimer's, including PICALM in this particular case, able to predict chronological versus biological age, and their differences to infer an aging rate, which is implicating very different pathways from ApoE4, and is also appeared to be acting synergistically with ApoE4. In other words, ApoE4 will have almost no effect if you're a slow ager, but with fast agers, we see this dramatic change in the impact of the ApoE4 allele on the odds ratio of Alzheimer's disease. As I mentioned earlier, we're looking also in exosomes and blood biomarkers of cancer, and also longitudinal progression of cancer. And lastly, we've basically developed a lot of high throughput assays for systematically testing these genes that we predict to be drivers of different disorders. In the case of schizophrenia, Maria Cusi in our lab collaborated with Kevin Smith and Kevin Egan at Harvard to basically systematically image the phenotypes of these knockouts of genes that are predicted to be drivers of schizophrenia, and to start modulating them in the context of these iPSC interventions. So collaborating with Wed9am, we're looking at these modular and programmable CRISPR-Cas9 and dCas9 systems that allows to activate enhancers or repress them, to edit individual SNPs, and to also knock out promoters or genes. And we're carrying out these changes in iPSC here. You're seeing that changes, for example, are leading to reduced expression of BIN1 by affecting these proximal and distal promoters. When we differentiate these cells into neurons, microglia, astrocytes, and oligodendrocytes, we can start seeing the cell type specific impact of these changes. And we are able to carry out these experiments at 384 well plate in parallel. We've also developed massively parallel experimental methods that allow us to test 7 million enhancers in a single experiment by cutting out DNA accessible sites, inserting them into plasmids that are self-transcribing, that allows to now test the expression, the ability to drive expression for millions of these fragments using these fragments as their own barcode. And then because of the random fragmentation nature of these regions, we're able to now start fine mapping the activity when it's coming from a region that overlaps these nucleotides. So in this particular case, we developed this Sharper2 tool that allows us to fine map the location of expression of these regions to evolutionarily conserved regions, as well as regulatory motifs, enabling us to now start fine mapping regulatory activity at very high resolution. We're using this to now start predicting individual variants that are likely to be causal associated with Alzheimer's disease, in this particular case in microglia. And in collaboration with Andreas Fenning, a former postdoc in my lab, who's now a professor at TMU, and our collaboration with the Cure Alzheimer's Fund Consortium, able to look at how these variants are acting in the context of mouse brain. So that's what I had to say in terms of understanding and manipulating disease circuitry at single cell resolution, looking at reference epigenomes, looking at how these vary across individuals in the context of genetic and disease variation, looking at single cell dissection, cell type specific genetic effects, multiple phenotypes, multiple tissues, and high throughput dissection. I'm very fortunate to be part of this incredibly collaborative team of scientists, both experimental and computational. We also have many partner labs, including Li Hui's lab, that we collaborate very closely with, collaboration with McLean Hospital and Brad Ruzicka, ROSMAP and David Bennett, and our funding is coming from the NIH and several funds, including the Cure Alzheimer's Fund. And lastly, many of the postdocs in our team have now gone off to start their own faculty positions at Columbia, McGill, Harvard, TMU, Stanford, UCLA, and so on and so forth. So we're looking for new postdocs. So if any of the trainees in this call are looking for either computational or experimental adventures in single cell dissection of disease circuitry, please let us know. I'll be happy to chat. All right, I'll stop there and see if there's any questions.
I'm ready to sign up and join you, Manolis. Thank you very much.
The feeling is mutual, Jonathan. I'd be happy to collaborate either direction. All right, so again, feel free to ask any questions in the chat or in person, and feel free to email me. I'll put my email address in the chat. So it's just manoli@mit.edu, and our website is compio.mit.edu.
People want to think about it, and they can get hold of you. And thanks for, a tour de force.
Thank you so much. I'll give you actually a link to the slides. Just 1 second. So for those students who want to go back, you can find my slides here. And I will make the recording accessible so that you guys can have any additional questions. All right. Happy to chat with everyone. Thank you so much. And then hopefully see you around Boston.
Thanks again.
Thank you, Jonathan.
Thank
Thanks,
you
Stephanie.
so much.
Of course, a pleasure. Thank you, guys. Bye.
Thank you.